Basics of Medical Biochemistry. First edition 2023. Chapter 16. Metabolism of Purines and Pyrimidines, Part 2 of 2. By Professor Dr. Muhammad Jawad. MBBS, Tao, M. Philosophy, Ph.D., Dip M.E. Head of Biochemistry Department-Fazaia Ruth Pfau Medical College Air University-PAF Base Faisal-Karachi-Pakistan. Ex-Professor of Biochemistry, Qassim University Kingdom of Saudi Arabia. Ex-Professor, Head of the Biochemistry Department and Vice Principal. Liaquat College of Medicine and Dentistry and Fatima Jinnah Dental College, Karachi, Pakistan. Chapter 16. Metabolism of Purines and Pyrimidines. 16.4 Synthesis of pyrimidines. Just like purine, there is no special dietary requirement for the synthesis of pyrimidines in the body. Body tissues can synthesize pyrimidines from the components of a normal diet. Pyrimidines are synthesized as nucleotides. There are two mechanisms for the synthesis of pyrimidine, one de novo synthesis and two salvage pathways. 16.4.1 De novo synthesis of pyrimidine nucleotides. It is a shorter pathway than purines. A pyrimidine ring is made first and then attached to ribose B, unlike purine biosynthesis. The required precursors are aspartate, glutamine and HCO3 for the synthesis of a six-membered pyrimidine ring. The final product is uridine monophosphate, UMP. Figure 16.7 Element source of pyrimidine base, ASP, aspartate, GLN, glutamine. Following are the steps of pyrimidine nucleotide synthesis. 1. Synthesis of carbamoyl phosphate. It is energy-consuming step. Carbamoyl phosphate for pyrimidine synthesis is made by carbamoyl phosphate synthetase 2, CPS2. This is a cytosolic enzyme, whereas CPSI is mitochondrial and used in the urea cycle and arginine biosynthesis. It is activated by PRPP and inhibited by UTP. Substrates required for the synthesis of carbamoyl phosphate are CoA2, glutamine and two molecules of ADP. Glutamine plus CoA2 plus 2 ADP, carbamoyl phosphate plus glutamate plus 2 ADP plus pi. This is the committed step in pyrimidine synthesis in mammals because carbamoyl phosphate made by CPS2 has no fate other than incorporation into pyrimidines. Defects in ornithine transcarbamylase of the urea cycle led to the increased synthesis of pyrimidine owing to the abundance of carbamoyl phosphate. Ammonia necessary for the formation of carbamic acid originates from glutamine. Figure 16.8 Structure of carbamoyl phosphate synthetase 2. Table 16.2 Comparison between CPSI and CPS2. Features. CPSI. CPS2. Site. Mitochondria. Cytosol. Metabolic process. Involved. Urea cycle. Biosynthesis of pyrimidine. Source of nitrogen. Ammonia. Gamma amide group of glutamine. Effectors. Activated by N-acetyl glutamate. Activated by PRPP and inhibited by UTP. 2. Synthesis of carbamoyl aspartate. Carbamoyl aspartate is formed by the condensation of carbamoyl phosphate with aspartate. This reaction is catalyzed by aspartate transcarbamylase, ATCase. Carbamoyl phosphate is an activated compound, so no energy input is needed at this step. Aspartate transcarbamylase is a classic two-state allosteric enzyme, ADP+, CTP. 3. Formation of dihydroorotate. In this step ring closure and dehydration takes place, catalyzed by dihydroorotase. 4. Formation of orotate. Synthesis of a true pyrimidine i.e., orotate takes place by oxidation of dihydroorotate catalyzed by the enzyme dihydroorotate dehydrogenase. This enzyme is a flavin mononucleotide containing protein found in the inner mitochondrial membrane. 5. Formation of orotidine 5 monophosphate. OMP or orotidylate orotate is joined with a ribose 5-phosphate to form orotidine 5 monophosphate. The ribose 5-phosphate donor is PRPP. The enzyme involved is orotate phosphorobosyl transferase. 6. Formation of uridine 5-monophosphate, decarboxylation of orotidine 5-monophosphate by OMP decarboxylase produces uridine 5-monophosphate. There is a single polypeptide called synthase that has separate catalytic domains for phosphorobosyl transferase and decarboxylase activities. Figure 16.9 De novo synthesis of pyrimidine nucleotides, FMN, flavin mononucleotide, CTP, cytidine triphosphate, PRPP, 5-phosphorobosyl 1-pyrophosphate. Synthesis leads to the formation of UTP and CTP. is sequentially phosphorylated to UDP and UTP. UDP UTP. ADP 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 ADP. Cytidine triphosphate, CTP, is produced from UTP by amination catalyzed by the enzyme CTP synthetase. Glutamine is required to provide nitrogen. CTP synthetase. UTP plus glutamine CTP plus glutamate. ADP ADP plus pi. Some of the CTP is dephosphorylated to CDP, which is then reduced by ribonucleoside reductase to DCDP. The phosphorylation of DCDP gives DCTP for the synthesis of DNA. The dephosphorylation of DCDP gives DCMP, which is then deaminate to dump. Formation of deoxythymidine monophosphate, DTMP or thymidylate. The immediate precursor of thymidylate, DTMP, is dump. The formation of dump occurs either by deamination of DCMP or by dephosphorylation of DUDP. Figure 16.10 Formation of deoxythymidine monophosphate, DTMP or thymidylate. Metabolic channeling by multifunctional enzymes of pyrimidine biosynthesis. Eukaryotic pyrimidine synthesis involves metabolic channeling, the transfer of metabolites between different enzymatic sites on a multifunctional polypeptide. There are 6 separate enzymes in prokaryotes and 3 multifunctional proteins in eukaryotes. 16.4.2 Regulation of de novo synthesis of pyrimidine. Biosynthesis of pyrimidine is regulated at ATCase in bacteria and at CPS2 in animals. Biosynthesis of both purine and pyrimidine are coordinately regulated in such a way that they are produced in equal amounts. Figure 16.11 A regulatory circuit that controls pyrimidine synthesis in E. coli. Pyrimidine antimetabolites. There are many pyrimidine derivatives such as 5-fluorouracil, 5-bromuracil, 5-ioduracil, and 5-iodo-2-deoxyuridine act as powerful antimetabolites. They can interfere with the formation of pyrimidines and nucleic acids. Other examples include arabinose cytosine, azidothymidine, AZT, and folic acid antagonists like methotrexate and aminopterin. These compounds have been used quite effectively for the treatment of viral infections, cancer, and AIDS. Defects in de novo pyrimidine biosynthesis led to clinical diseases like orotic aciduria. It is due to a defect in phosphorobosyl transferase or orotidine decarboxylase. Symptoms include anemia, growth retardation, and increased orotic acid excretion in urine. Treatment includes uridine administration. 16.4.3 Salvage pathways for the synthesis of pyrimidine nucleotides. Pyrimidine bases are salvaged to nucleosides which are then phosphorylated to nucleotides. By the salvage reactions, many nucleosides such as uridine, cytosine, deoxycytidine, and thymidine are converted to their corresponding nucleotides, i.e., CMP, DCMP and TMP respectively. Similarly, orotic acid is converted to orotidine monophosphate, OMP or orotidylate, and DCDP is phosphorylated by ADP to DCTP. Formation of deoxyribonucleotides. The deoxyribonucleotides found in DNA are derived from respective ribonucleotides by the enzyme called ribonucleotide reductase, ribonucleoside diphosphate reductase. The conversion reactions involved the reduction during which 2-carbon of diribose having a hydroxyl group, O, is changed to 2-deoxy derivatives. Ribonucleotide diphosphates like ADP and GDP are the sources to produce deoxy-ADP and deoxy-GDP. ADP plus NADP plus H plus deoxy-ADP plus H2O plus NADP plus GDP plus NADP plus H plus deoxy-GDP plus H2O plus NADP plus. Thioredoxin acts as a hydrogen acceptor in the above reactions in association with the enzyme thioredoxin reductase. 16.5 Catabolism of pyrimidines. In some organisms, free pyrimidines are salvaged and recycled to form nucleotides via phosphorybosyltransferase transferase reactions. In humans, however, pyrimidines are recycled from nucleosides, but free pyrimidine bases are not salvaged. Catabolism of cytosine and uracil yields alanine, ammonium ion, and Co2. Catabolism of thymine yields aminoisobutyric acid, ammonium ion, and Co2. These are highly soluble products. Figure 16.12 Catabolism of pyrimidines. Summary. Purine and pyrimidine are nitrogen-containing heterocyclic, aromatic compounds. De novo synthesis of purine nucleotides includes a series of complex reactions. The first purine synthesized is inosine monophosphate, IMP. IMP is regarded as a parent purine nucleotide that is rapidly converted to AMP and GMP, as it does not accumulate in cells. The most important regulatory step for the de novo synthesis of purine nucleotides is the first step, which is catalyzed by the enzyme glutamine PRPP aminotransferase. This enzyme is inhibited by AMP and GMP while activated by PRPP. Purine bases created by the degradation of RNA or DNA, an intermediate of purine synthesis, can be directly converted to the corresponding nucleotides by salvage pathway. The catabolic process of purine-containing nucleosides starts from adenosine and guanosine. These nucleosides are derived from respective nucleotides which are released from the breakdown of RNA and DNA. Adenosine is obtained from AMP by the enzyme 5-nucleotidase. Adenosine is converted to inosine by adenosine deaminase, a nucleoside deaminase. The ribose of inosine is then removed by purine nucleoside phosphorylase to form hypoxanthine. Hypoxanthine is oxidized to xanthine and then finally to uric acid by xanthine oxidase. The end product of purine catabolism in man is uric acid. The normal range of serum uric acid level in males is 3.5-7 mg/dl and in females is 3-6.5 mg/dl. Hyperuricemia is the condition of increased, above 7.0 mg/dl, levels of uric acid as urates in the blood, accompanied by increased excretion of uric acid in the urine. Gout occurs from the accumulation of uric acid crystals in the extremities, especially in the big toe of the foot. Lesch-Nyhan syndrome is caused by a severe deficiency in HGPRT activity. Pyrimidines are synthesized as nucleotides by de novo synthesis and salvage pathways. The formation of carbamoyl phosphate is the first step in pyrimidine synthesis. It is energy-consuming step and is catalyzed by carbamoyl phosphate synthetase 2, CPS2. UMP synthesis leads to the formation of UTP and CTP. UMP is sequentially phosphorylated to UDP and UTP. Cytidine triphosphate, CTP, is produced from UTP by amination catalyzed by the enzyme CTP synthetase. Glutamine is required to provide nitrogen. The formation of DUMP occurs either by deamination of DCMP or by dephosphorylation of DUDP. The biosynthesis of pyrimidine is regulated at ATCase in bacteria and at CPS2 in animals. There are many pyrimidine derivatives such as 5-fluorouracil, 5-bromuracil, 5-ioduracil, and 5-iodo-2-deoxyuridine act as powerful antimetabolites. Pyrimidine bases may be salvaged to nucleosides which are then phosphorylated to nucleotides. Catabolism of cytosine and uracil yields alanine, ammonium ion, and Co2. Catabolism of thymine yields aminoisobutyric acid, ammonium ion, and Co2. References. Exercise questions. For MCQ 1-5 see Part 1 of this chapter. MCQ 6. Which of the following molecules is a direct precursor for the pyrimidine ring during its synthesis? 5-phosphorobosyl 1-pyrophosphate. Aspartate. Fumarate. Glutamate. Glycine. MCQ 7. A 35-year-old man was brought to the hospital for constant severe pain with warm swelling in his right ankle for the last 24 hours. His family history revealed the same problem in his uncle. Serum uric acid concentration is increased. Which of the following enzymes is most likely deficient in this patient? Adenine phosphoribosyltransferase transferase. Carbamoyl phosphate synthetase 2. Hypoxanthine guanine phosphoribosyltransferase transferase. Ribonucleotide reductase. Thymidylate synthase. MCQ 8. Urinary excretion of which of the following substances is increased in the patient of gout taking allopurinol? Glycine. Inosine. Uric acid. Uridine. Xanthine. MCQ 9. A 46-year-old man recently experienced severe joint pain in his big toe, which was red, swollen, and extremely painful. His doctor suspects it might be related to uric acid levels. What is the most likely condition causing this symptom? Gout. Osteoarthritis. Osteomalacia. Osteoporosis. Rheumatoid arthritis. MCQ 10. A 32-year-old woman has been following a diet high in red meat and seafood for an extended period. She visits her healthcare provider for a checkup and mentions her diet. Her doctor is concerned about her uric acid levels. What dietary advice is most appropriate to lower uric acid levels? Avoid green leafy vegetables. Consume more sugary and carbonated beverages. Include more dairy products in her diet. Increase consumption of red meat and seafood. Reduce alcohol intake, especially beer. End of Part 2 of 2 of Chapter 16. Thank you.